Hey everyone. I hope your weekend was not horrible. Here's some like traffic or something. MIT's 1,000 degrees. Okay. Let me pull up the slides. All right. Final day of alcohol lectures. The comment box. Chat box is open. And we'll get through the rest of alcohol today. I don't know how long we're going to go. Maybe it's short, maybe it's long, whatever. We've done a couple of long lectures, so at some point I'll try to balance that way, cutting a couple of calories out of a lecture to try to balance the timing. But so just one more round through alcohol. That was traditionally my favorite gin. This is an alcohol church key. I think it's defunct. I don't think it exists anymore. But one of my old friends from, I don't know, middle school and high school and whatever, Jeremiah Hawkins, his brother invented church key with somebody from that show Entourage. I don't think it exists anymore. It was like a super, super hipster beer. But all of these different alcohols, when we're looking at how these things are formed, right, are ethanols, hopefully there's no methanol in there. We'll talk more about methanol today. That we're going to get this sort of microbe feces is where we're going to get this from, where we're going to get our ethanol, Saccharomyces. And this it's like brewer's yeast, right? This single-celled fungus is this little microorganism is going to shit out your ethanol, and that ethanol is going to give us all of our drinks, right? All of our alcohols. You remember this, just we've been trying to store and produce and consume alcohol for thousands of years, 10,000, 11,000 years ago. We're seeing all these pots that have alcohol residue on them. It comes from fermentation. Now in a human being, we get to pyruvate, and what do we do with it? Well, there's a couple of possible fates, but lactic acid, if you're being very glycolytic in your metabolism, acetyl-CoA if you're being very aerobic in your metabolism. But if you are yeast, right? If you are going to create your ethanol, what you convert the pyruvate into, you go through acetaldehyde and then to ethanol. And in us, we do sort of the opposite direction, but we don't end with pyruvate when we're metabolizing this. When we put alcohol in our mouth, when we put ethanol in our mouth, we use alcohol dehydrogenase, right, to convert into acetaldehyde, and then acetaldehyde or aldehyde dehydrogenase to dispose of it. And so again, fermentation in yeast, we're seeing that alcohol dehydrogenase enzyme is how the yeast is going to produce the ethanol. And that is our enzyme for disposal of alcohol as opposed to creation. The reasons that we have, or that yeast is going to ferment in this way, is going to create a bunch of ethanol. I don't know the why, right? We can do how questions pretty effectively, the mechanisms of things, but the why questions, that's speculation, has a little bit of, that's a loose waistband. I mean, you can really stretch that out and kind of fit it over just about any form, just about any supposition can put those PJs on. So, again, this is fungus. It reproduces asexually. That's what those little green, bulby, zitty looking things were. Carbon compound, alcohol is. There's your ethanol, and here's your methanol. Notice the one carbon versus the two. This is the one you don't want to drink. We've all had some. we all have a little bit of methanol now and again. It's in a very small dose. Ethanol is the alcohol we try to drink should we attempt to, you know, get a buzz or whatever. Ethanol is what we want. There's a hydroxyl group in ethanol, acetic acid, vinegar, right? There's, if we're talking steroid alcohol, cholesterol, steroid alcohol, right? We're seeing that hydroxyl group there as well. You remember what distillation is. We are heating that water up to try to get that volatile ethanol out of it, and then we'll isolate and condense it. The azeotrope, right? The limit 95.57% alcohol, meaning what's coming up in the vapor is the same composition as what's left in the still. So you can't distill higher than that. You can distill to a pretty high alcohol. You don't want to drink that. That's not for drinking. That's for like killing. I mean, that's like cleaning and killing and stuff. Again, there's your ethanol, and that's what methanol kind of comes first, right? You're going to get some methanol out of there. That's not what you want to drink. Now, as soon as you do drink this stuff, as soon as you do drink alcohol, gastric emptying is going to be affected. Alcohol itself does not empty from your stomach and find itself in circulation as fast as water does. Water, if you drink that, you drink 8 ounces of water or whatever, that's going to empty from your stomach faster than water with 4%, 10%, 40%, getting pretty high there. As soon as you start lacing that water with a little bit of booze, you're going to slow gastric emptying. It's not going to hit your system quite as fast. Anything you're eating with alcohol is also slowed down. So the gastric emptying of food, of whatever it is you're eating, it's like pretzels and nuts, or it's lasagna, or whatever it is you eat with alcohol, the barbecue, all of those calories are going to hang out in your stomach a little bit longer in the presence of alcohol. If it's a high calorie meal, if it's a low calorie meal, those meals kind of stay put in your stomach for just a little longer. And then the more food you consume with your alcohol, if you just have alcohol by itself, take your shots, go to the bar, eat none of those pretzels, and just have the alcohol by itself, it's going to empty pretty quick. Not as fast as water, right? But it'll empty out of you pretty quick. And as soon as you start putting in food, you're going to, it's not going to get up, it's not going to hit you quite as fast. You will slow the release, and maybe that'll give you a chance. Maybe that'll give you a chance to buffer a little bit of it if you can use the alcohol dehydrogenase in your stomach. If you're slowing gastric emptying, if you can use some of that alcohol dehydrogenase in your stomach to buffer some of it before it gets into your systemic circulation, crosses the blood-brain barrier, and interacts with like everything ever. But all of those rates of emptying can be affected, but once you start breaking this stuff down in your stomach, in your liver, right? You have your alcohol dehydrogenase. That's the main enzyme that you do your dehydrogenase, hydrogen acing to, and then acetaldehyde, dehydrogenase, aldehyde is sort of like the parent class of dehydrogenases, but aldehyde or acetaldehyde, dehydrogenase, and then you wind up with your vinegar over here, and that's your disposal. Now, again, if you're just tons of this stuff, you're going to come up with some more ways. You're going to hire some more, seasonal staffers to try to help dispose of it. But if you drink a bunch of alcohol, some of it, especially people who have a history of alcohol consumption, younger males tend to have a little bit more in their stomachs, and people who specificity of adaptation, right? If the stressor you're applying to yourself is ethanol, you're going to respond to be able to tolerate that particular stressor. You won't respond to be, okay, I got pretty good at holding my liquor. That means THC, or that means whatever, heroin I'm good at. Specificity of adaptation here. Alcohol dehydrogenase works on alcohol. but so you can metabolize a fair amount in the stomach before it has a chance, before it just bears a chance of crossing a blood-brain barrier, right? We're still in the stomach here, with the alcohol dehydrogenase living there, and this is men and compared to women across the lifespan. Younger men tend to have more. This is gastric alcohol dehydrogenase. Men tend to have more, women tend to have less. Later in life, you sort of hit that halfway point. It's like, who cares what your gonads look like? You got the same thing for your alcohol dehydrogenase in your stomach. Now this is just men over here. This one is just men, and under 50, right? All right, we got a bunch. Over 50, we've lost a lot. We're losing a lot here, and alcoholics tend to have less. And so alcoholics early in life, early in, let's call it training, early in training, when those drinks are coming in with regularity, you're increasing the amount of alcohol dehydrogenase you have. Later in life, you've overdone it a little bit, you've overtrained, and now as a consequence, a response to, or a consequence of that overindulgence, over ingestion, overtraining, you are going to have lower alcohol dehydrogenase activity, and you're going to be buzzed for a while. The flush syndrome, Asian glow or whatever, flush syndrome. Remember, this is that acetaldehyde or aldehyde dehydrogenase, where you retain the acetaldehyde. You'll dispose of it eventually. You don't just keep it forever, but this step, there's a holdup at this step. You get here just as effectively and fast. You cross the first finish line right there with everybody else, with alcohol dehydrogenase creating your acetaldehyde, but disposing of the acetaldehyde, that's where it gets a little bit tricky. And so you have this polymorphism. There's a genetic difference in the coding of that enzyme. Just one swapped amino acid, right? It's just glutamine lysine. You just swap one, and you get this sort of terrible form of that enzyme, and that's what the flush is going to come from. Prolonged exposure to acetaldehyde, and yeah, maybe 8% of the people, give or take, in the world have this. So it's not like, uh-oh, I have this thing and I see epidemiological correlations that say esophageal cancer. We're like, okay, that's like 8% of the world. There's nothing to even have one heartbeat of stress about, one anxiety pulse over that like epidemiological data says something about, like, hundreds of millions of people. Don't worry about anything like this. Just the idea of what this is, how it's metabolized, how we get our Heinz vinegar over here, or acetate, right? So the acetaldehyde dehydrogenase is supposed to dispose of the acetaldehyde, and if you have this lysine as opposed to glutamate version at that one site, single nucleotide polymorphism, one little site, you will do a terrible job of disposing of it. And so the acetaldehyde builds up. That's where you're going to get that flush syndrome. That's the easiest test to see if you have the glow. It's just, let's drink and look at your cheeks. So then just, pharmacokinetic versus pharmacodynamic, or metabolic versus functional. This is really what we talk about, right? The changes that affect the clearance of alcohol, whether it's the breakdown, let's build up some more alcohol dehydrogenase in our stomachs, what's happening in the liver, stuff like that. So on Friday, we talked about metabolism, metabolism, right? The balance between anabolism, buildup, and catabolism, cata, down, right? Ana, up, cata, down, metabolism. After all of your ana-ing and cata-ing, after all of your building up and breaking down, what's the net summation of chemical reactions and that balance between buildup and breakdown? Metabolism, right? And a real major hub of metabolism, of that balance between buildup and breakdown. Complex 1, mTOR complex 1, the mTOR enzyme, it's a kinase, an ACE, an enzyme, a kinase attaches phosphates to things, and mTOR phosphorylates a couple of things. P70S6K, very anabolic. That also is a kinase. It phosphorylates ribosomal protein S6. The ribosome is where translation happens. Translation, the making of proteins, the synthesizing of proteins, translating proteins. That's a ribosomal activity. And so as soon as you phosphorylated ribosomal protein S6, okay, let's turn the stove on, let's get the fires going, let's start cooking up some proteins. mTOR also, the mTOR enzyme, the kinase, also, the mTOR complex 1, also phosphorylates 4-EBP1, and that's withholding translation. 4-EBP1, that's an off switch on translation. And so when you phosphorylate it, when mTOR phosphorylates it, you shut off the off switch, right? So you withdraw inhibition of translation. So you phosphorylate one thing and turn on translation, turn on protein synthesis. You phosphorylate another thing and you turn off the inhibition, you turn off the policing of translation. So mTOR complex 1, if this thing is on, if this thing is doing its kinasing, if this thing is phosphorylating stuff, you are anabolic. You're in anabolic mode. This is so expensive, though. So expensive. I mean, this is like the biggest budget you have. I mean, this is really expensive for a cell to be translating proteins. Massive budget. And you could make an argument about AMPK. I'll show you that in a sec. We talked about it on Friday. AMPK also being the sort of this critical regulator of metabolism, the hub of all metabolism or whatever. AMPK, fine, but AMPK is interacting through mTOR in part. It is inhibiting mTOR. Through tuberin, which is upstream from just upstream from Reb, and through Raptor, regulatory associated protein of TOR. Raptor is a thing that recruits the downstream targets, so mTOR can actually phosphorylate them, so you can do your hypertrophy. So AMPK, mTOR, critical hubs of metabolism, massive critical hubs of your body's metabolic state, whether you're in anabolism mode or catabolism mode. Now, when we talk about, catabolism and breakdown, stuff like this, yeah, we'll talk about sort of classic definitions of what metabolism are, right? Glycolysis and the electron transport chain and Krebs cycle, and what are we actually, how do we burn our calories, beta oxidation, lipolysis and beta oxidation and whatever. Yeah, how do we burn our calories? That's just catabolism, right? That's a piece of metabolism, but that's catabolism. But the balance of anabolism and catabolism, that's metabolism. And where does that balance lie? What enzyme can we point to and say, you're the linchpin or whatever, you're the one, you're the team captain? mTOR is a reasonable candidate to point to and say, this is the foundation, or the like the rebar of metabolism, something like that. So upstream from mTOR, tuberin right here, tuberous sclerosis complex. TSC2 is tuberin. That's tuberous sclerosis complex 1, tuberous sclerosis complex 2. This is tuberin. This is the thing PKB, protein kinase B, or AKT, it's often called, AKT or PKB. That's the thing that it shuts off. That PKB, super anabolic, shuts off tuberin. And tuberin is facilitating the hydrolysis of Reb's GTP, right? So let's convert it to GDP. Reb GDP doesn't turn on mTOR. Reb GTP turns on mTOR, just like you can go from ATP to ADP, right? Adenosine triphosphate to adenosine diphosphate, 3 phosphates to 2, guanine triphosphate, guanine diphosphate, right? So that's what tuberin is doing. Let's shut let's hydrolyze this GTP. But if you shut off tuberin, Reb binds to mTOR, turns it on. Here's Raptor. Raptor's the thing that recruits those downstream targets so mTOR can phosphorylate them. And now we have protein synthesis or protein translation or hypertrophy or whatever you want to call it, depositing proteins, creating and depositing proteins in a cell. So we see the same stuff up here, AMPK, that's what I was talking about earlier. PKB or AKT, just talked about a minute ago. AMPK is activating tuberin. PKB is deactivating tuberin. So balance, right? Balance for metabolism. And there's Reb with the GTP, turn it on, mTOR. Remember mTOR, mammalian or mechanistic target of rapamycin, right? Here's rapamycin. It's an exogenous chemical, this macrolide, a big chemical. And FKBP12, that's where it interacts in the mTOR complex. But who cares? Who cares about any of this stuff? This is the only stuff that really matters is down here. And so mTOR complex 1, there's P70, S6K, sometimes S6K1. Again, these things have a bunch of names and it gets a little bit obnoxious. Ribosomal protein S6, you'll usually see a little RP before that, ribosomal protein S6. And so this, we're shutting off 4-EBP1, we're turning on P70S6K, and through both of those, we phosphorylate. mTOR phosphorylates both of these, shutting this one off, turning this one on. Protein synthesis is the consequence of that. Now, you take some alcohol, you put some, I don't know, brandy in your coffee or whatever. You put like a rum in your tea, whatever it is that you do. And we see a decrease, ETOH, that's ethanol. The control, this isn't like concentric, right? And some like weird spelling of like eccentric loading or something. So control compared to ethanol at 30 minutes post-contraction. So this is stimulation. This is non-stimulation. Ethanol in each one of these time points, now 12 hours post-contraction, you shouldn't have a blood alcohol content anymore at this point, but there's still a marginal depression of protein synthesis after 12 hours, after half a day. You wake up at 8 a.m. in your coffee, you just can't confront the day. So you put some, I don't know, vodka in your coffee or rum in your coffee or something. And then that's 8 a.m. right at 5 p.m. you go exercise, you know, go do your weights, and you may still have an effect. I say may because this is one study right here. And the conditions of how do you elicit this effect? Well, it's not by having, you know, rum in your coffee at 8 a.m. and then go doing a bunch of heavy resistance training at 5 p.m., right? That wasn't the experiment. But there is evidence that when you have ethanol, there may be, you know, 12 or so hours where you see a suppression of mTOR activity. You see a suppression of protein synthesis, a suppression of hypertrophy, of repair and regeneration of muscle. Your recovery from the time you exercise until tomorrow when you get back on the tennis court or you get back on it's volleyball, it's you know, track, whatever it is. You beat your body up a little bit on Monday. You go back to the court or the track or the diamond or whatever if it's baseball. You get back on the field and on Tuesday, and are you recovered? Part of recovery is avoidance of alcohol during these kind of peak periods. You're going to have a harder time recovering. Now, this is phosphorylation of some specific things that we're looking at. S6K1 or P70 S6K, phosphorylation of that. Remember, that's immediately downstream from mTOR. mTOR itself, and then PKB, AKT or PKB, a couple of different sites, and the control versus ethanol. And this is like, I don't know, there's nothing there to really notice. But it started with mTOR and PKB. Okay, we start to see a little bit lower. Okay, but now let's look after exercise, stimulation, right? After exercise, what do we see to P70SSK? Well, it goes way up with control with no ethanol. It goes up a little bit with as opposed to non-stimulation, right? Non-stimulation compared to stimulation, okay, there's a little bit, but it is not what you're seeing with control. And then some of these other ones, you start to see a much bigger depletion. Looking at the same stuff over here, there's ribosomal protein S6, the controlled stimulation. That's where you're getting the most RPS6 phosphorylation. Remember, ribosomal protein S6, the ribosome is where translation's happening. This is the last stop before you're linking up some amino acids and cooking up some proteins. S6K1 or P70S6K, that enzyme that phosphorylates ribosomal protein S6, you're seeing the same thing. You would expect to see those relationships because this is the thing that activates that thing. Same article, just talking about it, just chatting a little bit, saying what is fascinating here is we expected to see, because lots of previous literature showed, but we expected to see that, in rested, unloaded, unmechanically stimulated tissue, ethanol suppressed mTOR activity. But in mechanically stimulated tissue, what we expected to see was mTOR still firing, right? Because you have integrin signaling, integrins, that transmembrane protein that lives in a cell membrane, crosses it. And transmembrane protein crosses the cell's membrane and reaches into that extracellular space and notices mechanical environments in that extracellular space, relays messages indoors through a thing called focal adhesion kinase, FAK, focal adhesion kinase. It's another kinase, it phosphorylates stuff, but when you do that type of cell signaling, you bypass some of the canonical signaling of mTOR, PI3K, PKB. You bypass that stuff. And so what they found in this article was interesting, in that you could bypass that suppression of mTOR by ethanol was not just the normal pathway people once thought. It's also suppressing other stuff through other means, suppressing mTOR. Now, this was a good article saying maybe that's a good thing, right? If we can use ethanol as an mTOR inhibitor, right? We can use ethanol as an mTOR inhibitor. Maybe there's something to add to chemotherapy and radiation and immunotherapy, and throw in a little like booze into that cocktail, something else to help ward off metastases and growth of these of the tumors, and so maybe there's something good here. This, you don't need to know about. It's just comparing of like, okay, we have an mTOR inhibitor here, this INK 128 ATP competitive inhibitor. It binds to mTOR's catalytic site. It's a potent inhibitor of mTOR, and who cares, right? The ethanol that they were looking at, what they found, it was fascinating of activating AKT, that's PKB, right? AKT or PKB, same thing. Activating PKB phosphorylation and mTOR complex 2 assembly. Now, mTOR complex 2, one of the things it activates, just like downstream from complex 1, what you're going to see is activation of P70SSK and 4-EBP1. Downstream from complex 2, you're going to see PKB activation. So it would make sense that these two would travel in tandem. Whether that's the mechanism that is like, well, there's more complex 2 activity, so we get more PKB. There may be more independent activity with the two. But the stuff that you see with ethanol, promoting, mildly promoting complex 2. Downstream from complex 2 is PKP. So mildly promoting that, but inhibiting somewhat potently complex 1. And maybe what they found, that maybe this is the primary explanation, but there seems to be an inhibition of raptor, regulatory associated protein of TOR, raptor. Remember, this guy recruits the downstream targets. Raptor recruits these so that mTOR can phosphorylate them. And so the disruption of a raptor association is going to have something close to catastrophic effects on mTOR complex 1. So that's one thing that they found. How the mechanism, how ethanol would obstruct mTOR complex 1 activity. And then they also found inhibition of the phosphorylation of 4-ABP1. Again, maybe that could just be like, well, if Raptor isn't recruiting, so sometimes these things overlap. Just talking about their 4-EBP1 um, inhibition there, but also the Raptor thing. Again, if there is disruption of Raptor activity, you have disruption of the whole mTOR complex. SG, stress granule, doesn't matter what that is. There may be long-term differences as compared to the acute effects of ethanol, right? Acutely, what do you see ethanol doing? Well, we see it increasing mTOR complex 2, and we see it inhibiting mTOR complex 1, and we have some mechanisms, and let's measure 12 hours later, we're still seeing this. That's all acute stuff. If somebody is an alcoholic, in this case, if a mouse becomes an involuntary alcoholic when it's 4 weeks old, which is a reasonable distance into its lifespan, I mean, it's just wasted all day. What you see is an expression of ribosomal protein S6 that's going to increase. And so there may be sometimes when you suppress something, you increase translation of that thing. You end up with a higher concentration to try to find balance, right? Regulation. Okay, this thing is suppressed. How are we gonna? Let's overdo it now a little bit. And so who knows really what happens in a person with chronic ethanol exposure as opposed to these acute episodes of ethanol exposure. I don't think this is characteristic of a human being who drinks a lot. This doesn't seem believable to me. Now, maybe it is, but I kind of doubt this is a clear picture of a human being, recreationally drinking very often. But they're all I'm using this for is to say we don't have the end of the story, right? We have the beginning of the story for alcohol and mTOR. We have the prologue, right? We're not at the, I don't know, where like the rising action, whatever, the friends and enemies, switch sides, and none of that stuff. We're really early in this narrative. Now, remember, resveratrol is that compound that's in wine and grapes. Obviously, if it's in the grapes, you're going to see it in the wine. Obviously, if it's in the wine, you're gonna have seen it in the grapes. But what resveratrol does is help activate AMPK, adenosine monophosphate, AMP, just like ATP minus 2 phosphates, right? AMP1 phosphate left. Adenosine triphosphate, diphosphate, you got 2 monophosphate, you're down to 1, AMP. And AMP activated protein kinase, AMPK, that is technically activated by a bunch of things, whatever, but LKB1 is this primary upstream activator. But how does LKB1 activate it? You got to bind AMP to it first. You have to bind AMP to AMPK to permit the activation by LKB1. Liver kinase B1. Ubiquitous, it's all over the place. It's not just liver. Liver kinase B1 is what LKB1 stands for, though. It's a kinase, right? And it's going to phosphorylate AMPK and promote it, turn it on. Now, once AMPK is on, resveratrol helps LKB1. It promotes the activation of AMPK via LKB1. That's what resveratrol does. It helps turn on AMPK. So drink your wine, eat your grapes, whatever, get resveratrol in you, take it by pill form, resveratrol. You're going to help activate AMPK. Once AMPK is activated, it is phosphorylating, it's a kinase, it is phosphorylating tuberin, it is phosphorylating raptor. Now, you can phosphorylate tuberin in multiple sites. You can phosphorylate raptor in multiple sites. Some of those sites turn it on, some of those sites turn it off. If you're going to phosphorylate tuberin with PKB, yeah, you're phosphorylating it in a way that turns it on. If you're going to phosphorylate tuberin through AMPK, you're phosphorylating in the site that turns it off. So AMPK turns on tuberin. Sorry, I don't know if I said what I said. Like PKB shuts it off, AMPK turns it on. If I said the opposite, this is what I meant. So AMPK turns on tuberin, PKB would turn it off. If you turn on tuberin, remember tuberin is breaking down Reb's GTP. So you have to shut down tuberin to permit Reb to activate mTOR. But also AMPK is turning off Raptor. And Raptor is one of the sites where ethanol seems to exert its effect, exert its inhibitory effect on complex 1, mTOR complex 1. Raptor recruits the downstream targets that the mTOR enzyme, the mTOR kinase, is going to phosphorylate, shutting this one off, turning this one on. If a Raptor can't do its job, mTOR can't do its job. mTOR needs its assistant. mTOR needs its Raptor. AMPK shuts it off. Ethanol seems to shut it off. AMPK also turns on protein degradation, ULK1 and forkhead FOXO. The lysosomal system, and the ubiquitin proteasome system. We'll talk about those in another class. All you need to know is protein degradation, right? We're breaking stuff down. We're in atrophy mode over there. Over here, we're in hypertrophy mode. We're growing, we're translating proteins. We're at the ribosome, translating proteins. And over here, we are degrading proteins. We are breaking proteins down. We're chopping them up into their component amino acids. Atrophy over here, hypertrophy over here. AMPK turns on atrophy, it turns off hypertrophy. mTOR just actually does the exact opposite with both. It shuts off atrophy while turning on hypertrophy. So mTOR is horrible for things like cancer. You don't want growth, right? You don't want this like out of control hypertrophy growth of these cells. It's wonderful. mTOR is wonderful for athletes who are trying to get back on the basketball court tomorrow. If they had a hard practice today, what you need to do is repair those cells. You need those cells to be ready to have the structural integrity to be repaired for tomorrow. mTOR is wonderful if you're trying to get faster. mTOR is wonderful if you're trying to get stronger. Ethanol, at least acutely, exerts an effect on mTOR. What seems to be at Raptor and what may be at 4-EBP1. And depending on the type of alcohol, maybe you're also getting some AMPK activation, which shuts mTOR down at both.
I have a question.
Yes.
I don't really know how to word it, but based on what you just said about the basketball example, how does that relate to recovery? Because like you know how you have off season and like people take breaks from their sports and stuff like that. Like how does that all work then with mTOR? If you were to take like a 2 week break or a month off, how does that
Where you just do nothing, or where you go start to exercise, where you go start
where,
doing.
where you do nothing? Like you know how that you have like a season and then athletes might take like 2 weeks off, because they've been playing for
Yeah,
5 6 months straight.
2 weeks is fine. You're not. If you are in at that point at the end of the season, you're probably at least in some sort of functional overreaching state. You need a little bit of time. Are you going to come back after 2 weeks? Are you going to come back stronger? Maybe it's possible, right? You're definitely not going to come back weaker after 2 weeks, at least noticeably, significantly weaker. But if it's like 2 months off, you're coming back weaker. I mean, that's just remember the body is really paying attention to the last few days. Things like altitude or heat or whatever, you can stretch that duration a little bit, but for the most part, your body, the rear view mirror doesn't go back that far. Your body isn't thinking like, what is it, March 20 somethings now? Your body isn't thinking like, well, what kind of resistance training did I do in February? Your body doesn't give a shit. Your body cares about what kind of resistance training you did, over the weekend and the second half of last week, maybe, depending on how much like inflammatory activity is still lingering around in your body, and you don't really pay attention beyond that. And so you're going to want, um, you will want as much. Uh, as much signal for hypertrophy, as much signal for muscular development and recovery as you can get, but you need to balance it with the breaks. And so 2 weeks, yeah, it's fine. You're gonna have eat and sleep and recover and allow it's this putting tissues back together, Humpty Dumptying yourself back into functional condition. That can take a little bit of time. Um, 2 months is way too much time. That's just being lazy. That's just like, well, you could have been a good athlete if you like worked harder. You gave up 2 months out of the year. That's a lot. And so finding the right amount of time to take off, that's a lot of what coaching should involve, is figuring out, based on whether it's fasted, resting heart rate in the morning, first thing before you get out of bed, performance metrics, and keeping your eye on your athletes and seeing what their performance is like, and monitoring sleep schedules and things like that when you can tell you're in some overreaching. But yeah, I mean, there's ways to turn on mTOR very effectively without having to beat yourself up. But if you think about, like, why do you see in cycling, let's say, like Tour de France cycling, uh, look up Floyd Landis, and he was banned for testosterone. He's not trying to get big, right? He's not like, well, the cyclists are trying to avoid getting big, right? Old tennis players, yeah, ask the old tennis players, and like they don't look like they even lift, right? They're on steroids. And why are they on anabolic steroids? Why are they doing testosterone? For recovery, right? Because you work really hard on Tuesday. How the hell do you get back on the court on Wednesday? Shit's hard, right? All this direction changes and the exhaustion, and like, oh man, the joints just can't take it. And so how do you maximize recovery? Well, nutrition, drugs, let's throw some drugs in there, some testosterone or something. Um, Sleep, a lot of these things, but a lot of it's just how do we maximize mTOR signaling? And I've sort of taken some highway exits off of your question, but uh, what you want to do is optimize recuperative power. And a couple of weeks is wonderful at the end of a season. A couple of months is making excuses for what should have taken 2 weeks. Okay, I'm reading the chat box. I'm confused. So why AMPK would turn on something that stops Reb, but also turn off Raptor? Don't those both count? Okay, so yeah, this stuff gets a little bit weird. Remember, Reb turns on mTOR. Reb is the direct ROS homolog enriched in brain, RHEB, Reb. Reb binds to, it's the direct activator of mTOR. It needs its GTP to do that. What tuberin is doing is it's hydrolyzing its GTP. So it prevents. Tuberin is a police officer that says, nope, you stay over there, you're incarcerated or whatever, you stay over there. So tuberin is policing Reb. As long as tuberin is on, Reb can't activate mTOR. As long as tuberin is on, mTOR is not going to go do a bunch of hypertrophic things. It's not going to go like initiate protein translation. So AMPK is keeping tuberin on, and AMPK, if that wasn't enough, let's keep tuberin on. Tuberin's policing Reb. Reb would be activating mTOR, but can't now. So if that's not enough though, then AMPK is like just as a secondary, as like an insurance policy. I am also going to prevent Raptor from recruiting its downstream targets. So it's sort of a double negative that doesn't make a positive. It's a double suppression of mTOR through both tuberin, keeping that on so it won't allow Reb to activate mTOR, and then also inhibiting Raptor to prevent it from recruiting mTOR's downstream targets. So AMPK, once that gets turned on, it double inhibits mTOR, while also sort of its other arm is while also turning on protein degradation. Okay, let's get into some less. Let me know if you have any other questions about like metabolism stuff, and otherwise we're going to get topical again. We begin and end sort of superficially with alcohol. Just interrupt me if you have questions. So vernacular, don't write this stuff down. Now we're just sort of chatting, and there'll be a couple of things to write down. I'll let you know something there may be an accountability for, but now we're just chatting about alcohol. So the vernacular, when you watch an old, it's Lord of the Rings or Game of Thrones or whatever, there's like ale that people talk about, and oh, that's different from beer, that's different from lager, and we don't really have a good distinction from these names anymore. And so originally, right, beer was made with fermented honey and fruit juice. It was an English drink. And ale is what we now think of as beer. A couple thousand years ago, is ale. Germany, right? I mean, like, well, but the names of these things, what people, when they try to get scientific with this stuff, they say, well, where does the yeast live? Right? We have yeast, Saccharomyces. Right? This is the one that is your brewer's yeast. This one. And Cerevisiae, that's the one. This is the one, right? Who cares about this one? This is really the brewer's yeast everyone uses. And we say if you lager, okay, that's at the bottom, right? The bottom fermenting yeasts, that's the lager. Ale yeast, that's at the top, but not really, right? That's when people are getting really kind of hipstery in their definitions, but the reality isn't really that. We just have regional terms for these things. And so, whatever, it's lager, it's ale, okay, it's all the same thing. Just what zip code are you drinking this in? That's really what these names come down to. American and German beers or lagers, English beers or ale. I mean, it's where what's your latest stamp on your passport? That tells you what you're drinking. The yeast isn't really important here in terms of the vernacular, but the flavor is fascinating because you get something like everyone knows tequila is almost impossible to drink unless it's really expensive. I mean, that stuff to me, I can't drink tequila. Super expensive tequila. Okay, that's different, right? That's a little bit different. Vodka tastes different from, I mean, gin, whatever, the juniper and stuff. But these drinks have very different flavors in them. So this idea of if it's just water and ethanol, why do these drinks taste so different? Alexa's saying tequila is your favorite drink. You must get like the good stuff. I formal. But when you look at some of this stuff, these other compounds that you might see in it, obviously the acetaldehyde, maybe there's a little methanol in there, and that hydroxyl group, right? If you're going to replace that with something else, these esters that we're seeing, these esters are usually derived from carboxylic acid and an alcohol, the COOH stuff. So there can be chemical differences in the ethyl acetate over here, the ethanol, carboxylic acid. But when you get into like vodka, what it's supposed to be is the purest drink, the purest alcohol there is, where it's nothing in there but water and ethanol. Nothing but water and ethanol. That's what vodka is supposed to be. Now that ratio can be a little bit different depending on the brand, depending on the whatever, the region and whatever. The percentage of ethanol can change. But water and ethanol with no impurities, that's the goal. That's what you want in your vodka. And yet there's brand loyalty, right? We see these differences, and oh, I love this one, I hate that one. I can blind taste test it and tell the difference. So this is from totally legit article, a legit journal. This isn't just like some drinker's fake thing. So vodka's definition, like neutral spirits so distilled or so treated after distillation with charcoal or other minerals as to be without distinctive character, aroma, taste, or color. The purpose of vodka is to be the white fish. You know, the fish that like doesn't have flavor on its own. You have to season it with stuff. It's like salmon tastes like something. Tuna tastes like something, right? White fish, a cod or whatever. That's just like, okay, that's ready for the sauce by itself. Nobody eats it by itself. And so that's what vodka is supposed to be. It's supposed to be like the white fish of alcohol. And yet we're seeing differences in flavor, even if something has the same ethanol percentage. We're seeing some differences in the flavor. So these clathrates, what this is if you get the alcohol percentage high enough, 44%, whatever alcohol, the water can create these cages, can create these they can crystallize and sequester the ethanol inside of it. And the differences of how many of these clathrates exist and the structure of this, maybe there's a difference in perhaps not the flavor of the alcohol. If you're just doing actual taste buds, there probably aren't taste buds for hydrogen bond strength or anything like that. So we're starting to tread into some speculative territory, but the mouth feel, right? How the alcohol is received in your mouth, that probably changes based on these structures. If there's low structurability, you're looking at a watery alcohol. This vodka gets a little bit watery if there's low structurability. If there's high structurability, you're getting uh, ethanol sequestered in those water cages, and maybe clusters of alcohol molecules start to appear. The chemistry of it starts to change, and that might change some of the mouth feel of the alcohol. So how do vodka drinkers develop brand preference? And so just talking about the structures of these things, again, maybe it's not flavor, maybe there's no actual taste difference, but the texture of that fluid could be a little bit different. I keep tripping over my plug. So then we've all had, like the black licorice flavor. So Ricard, if anybody wants to do it in like a French accent. This is what replaced absinthe, when absinthe was for a time made illegal. Late 19th century France. People who were drinking absinthe were just wild behavior. It's like, oh, it's a hallucinogen, and we're getting these wild and violent behaviors, and absinthe was made illegal. And you've probably had absinthe before, and there's a couple of interesting things about it. One of them is that black licorice flavor, and then the other one is it goes from translucent to opaque, right? You pour water in it. You have two clear liquids, and you pour the one clear liquid, water, in the other clear liquid, right? The alcohol, and it like becomes milk. Now it doesn't taste anything like milk, but But what is causing that? And sort of the history of this stuff is a little bit fascinating because what was in absinthe that people were so worried about was wormwood. Right, wormwood is what it's a hallucinogen in massive doses, but you're not going to get it in a massive dose. It's really not dangerous. Wormwood contains this thing called thujone, thujone, and that is what we think of as the hallucinogenic properties of absinthe, which was making people go wild and but like sage, the seasoning of the plant, whatever, sage has more thujone in it, right, than wormwood does, and sage is totally unregulated. Nobody cares about that. So the reason with absinthe that people were going crazy and it was such a kind of fascinating drink is the alcohol percentage was like 70 to 80%, not proof, but percent. Right. And so it was just outrageously high alcohol. People were just super drunk. It had nothing to do with the absinthe itself. But that was made illegal. And then this came along in its place and had that black licorice flavor. And with that black licorice flavor, it's the anethol. This is the thing that makes it go from clear to milky, cloudy, opaque, whatever. Now, water has a high surface tension. The surface tension of water is high. If you get water droplets to touch, bring those water droplets together, they merge with each other. I mean, the water droplets just merge with each other. Soap bubbles, that's a low surface tension. Yeah, maybe they'll kind of hook up on the sides, but they're not going to merge in that same way. Anethol, um, there's a low tension too. And when you pour water in with the high tension, you get this emulsification, you get this emulsifying. And there are actual legitimate articles that talk about this, that talk about the emulsification of anethol and how this works. And it's what's fascinating is like if you take salad dressing and shake it up, whatever, the vinegar and the oil, yeah, it lasts a little bit. Even something like coffee, leave that out for a long time and you're gonna start to see separation. But this, you know, your Ricard, if you have this and you pour the water in, that's like months. That stuff is going to hang on. I mean, that stays emulsified for a really long time. So it's a fascinating chemical reaction and a delicious drink if you like black licorice in liquid form. Okay, so let's get on to Visalgia, or the hangover. I think everybody has experienced this in some intensity or another, the hangover. You wake up the next day and you just want to go right back to sleep until the next day after that. Whether it's nausea and gastrointestinal discomfort, it's the headache, it's the exhaustion, maybe there's light sensitivity, whatever it is. This hangover, we've all experience it. So Visalgia is the medical name of it, but Elpenor syndrome is another medical-ish name for it's often used to characterize a really severe hangover as Elpenor syndrome, but also sort of the psychiatric disorder of extreme drunkenness where there's this deranged behavior. Elpenor syndrome describes that too. And it's not just like a social networking term. I mean, you'll see it in the literature as Elpenor syndrome. But so this is a guy who survived. The reason Odysseus is here, Iliad and the Odyssey, right? Elpenor is in the Odyssey and he survives the Trojan War. And then he's super drunk and he climbs up onto Circe's roof and falls asleep. The next morning, on the palace roof, the next morning, I think he hears his name being called. He hears some commotion, whatever. He wakes up and forgets he's on the roof and goes tumbling off, and he like breaks his neck and he dies. Right? Like Odysseus can't be bothered to find him. It's like, you know what? We got to set off, and they go to Hades, and like the first shade or ghosty thing, whatever, they get to, is Elpenor. It's like, hey, I died. You got to go back and find my body and bury it. Right. So there's the Odyssey in the good parts version. Whatever. He goes back and buries him. Uh, but Elpenor syndrome can be used to characterize a very severe hangover or the sort of deranged activities and behaviors of somebody who is in the middle of the acquisition of a hangover. This is sort of an older word, philoglobalized, right? It means that like you're trying to pretend like you don't have a hangover. One of my best friends in my undergrad, one of these sort of dearest friends I still carry in life, is we went to undergrad together, and we went to Pacific. We did our masters together too. But he came to class one day, it was ex-phys or something, and vomited all over the wall. And as he's coming in, and the professor, Peter Harmer, is like the most famous professor in the entire school. I mean, the guy publishes in New England Journal of Medicine and JAMA and stuff. And so my friend I don't want to say his name unless I already did, but he comes in and just vomits violently on the wall and is like sweating, and Peter's like, "You look ill. I think it'd be best for your mental and emotional and physical health if you just slept this one off. It's okay. Totally excused absence. I approve of the effort of trying to show up in this condition." And Peter probably thought he had some sort of flu or something, but he was just super hungover. That was philoglobalized. That's like showing up and pretending like, what are you sick with? Well, it's not a hangover. It's a condition. It's something else. Um, but getting your blood alcohol content over about 0.1, 0.08 is sort of that magic space where you're getting into your zone, into your little special, enjoyable kind of where you rejoice in everything space. Above 0.1, and with few exceptions, you're probably going to have some dimension, some capacity of a hangover the next day. It's going to take a little while to get there. There's just no evidence that it's withdrawal. Some people will say, like, what is the hangover? Well, it's alcohol, withdrawal. It's like this withdrawal symptoms. Are like, that's not it. Right? That's not causing a hangover. That's what's causing somebody who's been an alcoholic for a very long time, who cold turkeyed it. That's those symptoms. That's what that comes from, not from acute hangovers. But dehydration, there's one explanation. So this is where to start paying attention a little bit again. We're getting out of the just sort of narrative topical alcohol stuff and getting back into some physiology. Alcohol blocks vasopressin. Remember, vasopressin's old name was anti-diuretic hormone. It inhibited diuresis, the loss of fluids. So vasopressin, anti-diuretic hormone, inhibits diuresis. If you block vasopressin with alcohol, you facilitate diuresis, you permit diuresis. So the trigger for its release, right? We talked about vasopressin earlier when we were talking about the kidneys and in the tubules of the nephrons, right, in your kidneys. Yeah, a little bit of vasoconstriction, but you're retaining water in the tubules, right? In the tubules, you're retaining water in your kidneys, and alcohol is going to block that. And when you block that, you're going to get a little bit dehydrated. You're going to pee more than you drink, depending on the alcohol percentage, right? If it's like 5%, whatever, you're fine. But if you're drinking whiskey, right, your vasopressin inhibition is going to dehydrate you. You don't see electrolyte levels differing all that much. The electrolyte levels of somebody who has finished drinking and entered their hangover, their vassalgia, you don't see major differences in electrolyte levels. So, but what people say, sort of fraternity wisdom of how do you cure a hangover. A lot of people say, oh, just make sure you get that Gatorade first thing in the morning. Wake up and just pound your Gatorade, whatever. It's dehydration, it's electrolytes, and you're going to rehydrate and get those electrolytes back in, and goodbye hangover. Right? That's a lot of the fraternity wisdom that I've heard over the years. My favorite quotation comes from Callie Van Valkenburg. She was one of my students in 2016, probably something like that. And when I was talking about alcohol in her class, she says, do you get hungover when you're merely dehydrated? And you can get just as dehydrated. You can get more dehydrated than you are when you're drinking, than you are when you're hungover, by just going and like exercising in the heat. And the consequence is not a hangover, right? Dehydration and hangover are two totally different sensations and have two totally different causes, right? Because you can rehydrate in somewhat of a hurry. You can rehydrate pretty quickly. We went over that, the gastric emptying rates and how quickly do you see it even entering your sweat, 10 minutes later, and you're seeing the water entering your forehead. And so, I mean, it doesn't take long to rehydrate yourself, and merely dehydrated, you don't get a hangover. And you don't really see differences in electrolyte levels of people who are hungover and people who wake up just normal. And so we can sort of rule those out, the dehydration. Now yeah, you will get dehydrated, and yeah, you should probably put it back in some fluids because dehydration is not helpful. But it's not the culprit that people have assigned it to be. Blood sugar, okay, maybe the dehydration that accompanies alcohol ingestion is going to reduce your blood glucose levels. Maybe we're going to affect hepatic glucose production. Maybe our blood sugar is going to go down a little bit. Maybe there's a little bit more reliance on ketones. Okay, sure, the blood is more acidic. But if you measure the ketone levels of somebody um, who's in the hangover or whatever, and you just measure mine now, mine are higher because I'm on a ketogenic diet, and I have been for like, I don't know, 2 years or something. And I fast on Sundays, and it's Monday. My ketones are probably outrageous. And I'm not hungover, right? No part of me is hungover. When I fast on every Sunday, I go like 36 hours without anything but water. And at no point do I, succumb to a hangover. And so this isn't a thing either. The idea of blood sugar and big ketone levels or whatever, that's not it either. So far we've ruled out dehydration, blood sugar, ketones, and your electrolyte levels. Now, there may be something here with what's called congeners. The congeners are these other stuff, right? So you see over here, there's the dark rum, and over here, there's the light rum, whatever. Fewer congeners over here, less of that stuff, less additional things other than water and ethanol in this drink, almost certainly, than there is in this drink. What's giving it all that color? The congeners, right? It's other stuff. So the darker the liquor, often you expect there to be more congeners than you see in the clear liquids. Now, when we get into something like wine, and people say, well, wine, oh, it's the red wine has sulfites in it. Red wine has sulfites as a compound with a sulfite ion, SO3. And people start blaming all this red wine. It's a nice little Scientific American bit. This isn't like a wonderful journal or anything, but they do a nice breakdown of the sulfites in wine. White wine has higher. This isn't causing the problems people think it is. In wine, in red wine, it's probably more about serotonin. Serotonin has so many different receptors. Most of this is in your gastrointestinal tract, it's like bowels. It's like getting your shit on the march. But serotonin has a lot of diverse effects, and that's probably closer to the problem people have with red wine. Histamine, though, is going to be in wine, it's going to be in beer, it's going to be in sake. So you're going to have some histamine. Maybe there's something there. Maybe there's a little problem there. People talk about the tannins. The word comes from tanning hides, the tannins. But nobody is getting these migraines when they eat pomegranates. Most berries and some nuts and chocolate and cinnamon and cumin and a lot of seasonings, lots of stuff. Go eat your chocolate and vanilla ice cream, and the chocolate and vanilla both have tannins. Lots of stuff is going to have this, and people are like, no, 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 the wine. Like, okay, well, but maybe we're just latching on to something that isn't quite supported, that doesn't you know that isn't quite as meaningful as we think it is. I'm not saying that tannins are not a problem at all, or sulfites aren't a problem at all. What I'm saying is they're probably not the problem that people think that they are, and serotonin may be the problem. So getting into, back into the generalities of hangover, maybe acetaldehyde could possibly contribute. Acetaldehyde does, the toxicity of acetaldehyde does produce similar symptoms to being hungover. You can like induce this, right? You sort of induce something similar to a hangover, just load them up on acetaldehyde. But when the hangover arrives, you don't really have a bunch of acetaldehyde. You've metabolized this stuff. This is well after the fact. And so acetaldehyde levels have nothing to do with hangover or hangover severity. Acetaldehyde is just like almost non-existent when you're experiencing the peak of your symptoms. And so acetaldehyde is an unlikely candidate as a contributor. Maybe it's a domino contributor where it sort of starts something early, it starts a process early, but itself is hardly culpable. I don't think it needs to be, sentenced if it's, put on court. Methanol is fascinating. Methanol, again, this often in bad alcohol, it tends to be higher. It's in tiny amounts in good alcohol, right? In lethal alcohol, it's in lethal amounts. That's why the alcohol is lethal, is you drink a bunch of methanol. Uh, and yeah, you'll get drunk early on. Early on, you'll have a similar effect of drinking ethanol, and then you'll start having the bad effects, right? So the alcohol dehydrogenase works on it just like it would same enzyme, just like it would ethanol. But instead of converting it into acetaldehyde, like it would with ethanol, you convert it into formaldehyde. Okay, formaldehyde is that thing that smells so horrible in a cadaver lab. Formaldehyde. That's not really something that you want inside of your body. I mean, the cadaver doesn't even want that on them, right? And so that's not something you want in excess of inside of you. Now, worse, the acetaldehyde or aldehyde dehydrogenase, you're converting it into formic acid. This isn't vinegar over here, right? That is ant venom. You're converting it into ant venom, not vinegar. And this is where, once you get to this point, you're having some pretty severe side effects. Again, right away, the methanol, you're going to get similar effects. You're not going to notice. You're not going to tell the difference. Maybe you can taste the difference. There's mouthfeel or something like that. This vodka is good. This vodka is bad, and you can tell because you're good at telling. Uh, but really, you're going to have the inebriation. You're going to have this stuff. But as soon as you start turning it into formic acid, right? Ant venom. This is an inhibitor of cytochrome C oxidase. That's the last enzyme in the electron transport chain. Now, the electron transport chain, this is how you use your oxygen. This is the same thing cyanide blocks, right? Cyanide, what people talk about killing people with apple seeds. Oh, I collected enough apple seeds to murder somebody, right? Cyanide can be derived from apple seeds, and cytochrome C oxidase is the same thing that inhibits. And this is the final step in the electron transport chain where you're actually using your oxygen. You are transferring the electrons to the oxygen. You're actually making water at this step. So metabolism, the oxidative metabolism is really disrupted. That's bad. Now this, you're really going to start encountering problems. Um, One of the first symptoms that you'll probably see is vision problems, right? So tunnel vision, loss of color, things like that. Methanol, those eyes, that's going to be a good indicator that that alcohol might have been a little messy, right? Maybe we should have bought more expensive, vodka. My eyes are getting weird. Not like I'm drunk and I'm staggering around and everything. The world is wobbly and I have to look at you with one eye. Not that, right? But this idea of there's a different kind of vision problem, and you would know if you had it. Now the cure is fascinating. It's not a good cure, but it's a solution of a kind. And go drink a bunch of good alcohol. You just drink a ton of terrible alcohol with a bunch of methanol in it. All right, here's the good stuff. Start drinking this. Let's try to overpower our BAC with ethanol, because then the alcohol dehydrogenase will start metabolizing the ethanol in the place of methanol. It will preferentially uh, metabolize ethanol and get our acetaldehyde, and your hangover is going to be horrible tomorrow. But let's try to limit the amount of formic acid that we're going to get, the amount of formaldehyde and formic acid that we're going to get. And then maybe you can piss a little bit out, you can breathe a little bit out, you can eject some of this without the creation of formic acid. So it's a nice solution, is just to go get wasted on expensive alcohol if you've just finished drinking some really terrible alcohol. Like, oh shit, my eyes, the emergency room is filled with COVID, I better just go get some really good alcohol. I don't assume this is like Courtney's solution to everything, but now the hangover, though, it's probably not going to be a bunch of methanol and formic acid and whatever, unless you really have problems. Really, what the hangover is caused by is a bunch of inflammation. Right now, cytokines. A cytokine is a signaling protein. A cytokine is autocrine, paracrine, endocrine. It's a cell releases this little protein. It's a small protein, a cytokine, a small protein that does signaling stuff, whether it's in the endocrine system circulating around all your systemic blood flow, or it's paracrine signaling, it's an adjacent cell. One cell releases a thing and it targets an adjacent cell, or it's autocrine, a cell releases a thing that then binds to itself. These cytokines, very immunological, immune system stuff. They're made by mast cells, they're made by macrophages, they're made by lymphocytes, endothelial cells will release it. Lots of things are going to make cytokines. And interleukins 10 and 12, interferon gamma, right? These are just a bunch of cytokines. Now, the interleukins, there's a bunch of these things. Interleukin 1, 2, 3, 4, 5, 15, 16, 17, 18. Right, there's a bunch of interleukins, and named in 1979, right outside of Interlaken, Switzerland. There was this conference, and the name, they say, has nothing to do with Interlaken, but often, if I name a bunch of like Tom Hanks movies, you're probably going to think of Tom Hanks if I ask you who an actor is, right? Old like magician tricks of, you just say a bunch of stuff that correlates with something, and then you ask like, what's your favorite sports team? And you just said 1,000 things about Denver or whatever. And so maybe that plays in, but really interleukin, leukocyte, between leukocytes. So these interleukins, interferon gamma, all of this inflammatory stuff, a bunch of cytokines, a bunch of inflammatory stuff get really elevated in the presence of your hangover. Now, can we treat it? Can we do something about that hangover knowing its cause? And tolfenamic acid, or tolfenamic acid, or whatever it is, I've only ever seen it in print. I haven't heard, this drug. It is still used. Now, this is a study from 1983, but if you fast forward the world to like now, you see this stuff being used for like a cancer patient is getting radiation, and let's see if we can make this a little bit more effective, and let's give them this tolfenamic acid. And so it's still used. This isn't some old weird thing, but more effective than NSAIDs. It's an inhibitor of prostaglandins. Now, NSAIDs, non-steroidal anti-inflammatory drugs, they're blocking COX, cyclooxygenase. They're a COX blocker, a cyclooxygenase inhibitor, and so you're inhibiting prostaglandins. This is just a really effective prescription version of that. And what they did was they gave 30 people. This is in the 80s. It'd be hard to rerun this exact experiment today as almost violently as they did with the blood alcohol contents. But they drank to 0.2. You get to 0.1, and you're going to have some symptoms tomorrow. You know how severe it is. It's up for I don't know about argument, but it's up for variation at least. 0.2, we're all hurting. This is painful the next day. I don't care who you are. This hurts the next day. And so people either got a placebo or sulfonamic acid. And then they drank after having this. Before bed, they took another. You know, if they were a placebo person, they took a placebo. If they were a drug person, they took the drug. And so they pre-medicate, then they drink, and they medicate again, and then they go to bed. When they wake up in the morning, the drug group is way better than the non-drug group. So prostaglandins, an inflammatory, it's an eicosanoid. It's made from fat. It's made from arachidonic acid. It's a 20 carbon like fatty thing. But you block this, and we actually see an improvement. Now, that's a drug. Can you go get something just from like the health food store or the grocery store or whatever? Yeah, it's not going to work as well, right? It's not going to work nearly as well, but there's some evidence. Great journal. Some evidence on hangovers of the prickly pear cactus. That's what this thing is. The abu tia, whatever. I have no idea how to say that. It's the prickly pear cactus. That's what colloquially it's what it's called, and so it's what I call it. So here, really good study, double blind, right? Placebo controlled crossover trial, which means the administrators and the subjects had no idea which person was getting which until all of the data had been analyzed. And then you reveal, okay, that was the control condition, and this was the prickly pear condition or whatever. And then they did it twice. And so people were their own comparison group. Really good study, this one was. And so 5 hours before alcohol consumption, again, pre-medicating. You're pre-medicating here. Once you've built up all of this inflammation, once it's already there, taking things that block its creation do you almost no good. They don't do you no good, but they do you almost no good. What you need to do is stop the creation of these things. And so you have to take them before you go drinking and at night before bed. You don't take stuff after you're hungover. It's already there. You're not curing the hangover. You're trying to prevent it. That's the effective strategy. And so then they go get them drunk, right? They do the pre-medicating, they get them drunk. 58% females, pretty well balanced, a bunch of, late 20 somethings, some 30 somethings, some early 20 somethings in there. These are normal drinkers. Hangovers per month. People are having, 1 to 2 hangovers per month already. These are actual, like, legit drinkers. And then here's a bunch of the symptoms of severity. And here's your prickly pear, and here's your placebo. And what you see is, the nausea much lower in the prickly pear, anorexia, whatever, much lower. So I sort of nutritional stuff, dry mouth, but the overall symptom index, the headache wasn't that much different, right? P is 0.34, meaning if there is no difference in real life, like if from Lord Almighty's calculation, there's no difference in here, and this is just sort of like sampling errors, how you found this, there'd be a 34% chance that you'd have observed an effect this big or bigger. That's what that means. And, so maybe there's something here with the headache, but there's some other stuff. Some components of the hangover are different. And again, here's the placebo and the prickly pear. But what you see is cortisol levels go up with the prickly pear. C-reactive protein that goes down, not down from baseline, but it's compared to the placebo group. Prickly pear is higher in cortisol and lower in C-reactive protein. Now, we talked about cortisol HPA axis. You drink and you get a bunch of additional HPA axis activity. You seem to get a little bit more HPA axis activity with the addition of the prickly pear. The C-reactive protein is important, though. This one is made in the liver. It's in systemic, it lives in the blood, systemic circulation. It's made in the liver, and it levels rise in response to an inflammatory increase, an inflammatory I don't know, system. And so stuff is released by macrophages, stuff is released by fast cells, and all this inflammatory induction is going to cause a rise in levels of C-reactive protein. And so this is an indicator that we are limiting the inflammatory response of alcohol with this thing. So if you want to create a new fraternity or sorority protocol for managing hangovers, manage them before drinking and before bed at night, and use this. Go ahead and throw in some NSAIDs just a little bit. I wouldn't use a bunch of Tylenol because you've already beat up your liver a bunch. You don't need to beat it up anymore. And aside from that, Tylenol is not an NSAID. But yeah, throw in some ibuprofen or something a little bit. Throw in some of this the night before, and that is the end of our alcohol session. Do we have questions?
Can age contribute to how hungover you get? Like, I feel like I'm 23 now, and I feel like when I was 21, I did not get as hungover as I do now.
Yeah,
So, like,
it'll get worse. Yeah. Yeah, the way we handle alcohol, it's the same thing as we're going to transition into sleep here pretty soon, and circadian rhythms and stuff. And it's the age just makes jet lag so much worse. A young person, you take someone who's like 12 or 15 or something, and just fly them all over the world. They sleep just fine. They sort of, you know, acclimatize to whatever the sun is doing, like immediately. I mean, just fine. And then take that same 15-year-old when he or she is like 45. It's like, man, it takes me like 3 weeks to adjust to this. It's like the heart, you're having like, heart problems and stuff after. And yeah, alcohol, you're seeing a similar effect where the hangovers tend to hang around perhaps a little bit more severely, and they overstay their welcome. It's like, oh yeah, this will be gone. I'll just have a little breakfast and it'll be gone. Like, what the hell is it still doing here? Like I did not extend an invitation to stay beyond breakfast. And so yeah, that happens. And there must just be I don't really have a good answer to it, but there must just be an inflammatory response that's a little bit more robust or harder to clear. Yeah, it'll get worse. Other questions? Okay. Well, I went over like 3 times in a row, so maybe I'll back off a little bit for a couple. If you have like a huge breakfast, just have a mellow lunch. All right, are we getting out of here? See everyone.